In this video, I'm going to review some of the basic genetics concepts that I brought up in lecture in the context of Hereditary Hemorrhagic Telangiectasia, or HHT. Oftentimes, in order to see a cell underneath a microscope, we have to apply some sort of dye to it. And in the early days of cell study, people were applying all sorts of different dyes to cells just to see what would show up. And early on, cytologists, people who studied cells, they noticed that there were these strange, weird-looking structures that would become stained inside of the nucleus when they applied certain dyes. And they couldn't simply call them weird structures, so they gave them a specific name, and they called them chromosomes. Now, this is a slight step up because, well, chromosome literally means colored bodies or colored things. Chromo meaning color, some meaning body or thing. And back then, they didn't know what these chromosomes were made of, much less what they did. We have a little better understanding today. And we know that chromosomes are essentially the packaged DNA of a cell. And we can group segments of it, segments of this DNA into genes. So what is a gene? Well, a gene, there are different definitions for gene, but one way of thinking about a gene is that it's a unit of inheritance. This is what can be passed down from generation to generation. If you want to be a little more technical, you can say that a gene is a DNA sequence that is a blueprint for a cell product. Blueprint. And I'm using the word product here very specifically because oftentimes whenever you hear about genes being talked about, people will say, well, genes code for proteins. And this is true. Genes do code for proteins, but they can also code for things like RNAs, which have particular functions inside the cell. So now we have genes, and we know that these genes code for cell products, and the genes reside on the chromosomes. Chromosomes themselves can be classified into two different types. One, you can have sex chromosomes. I'm going to abbreviate chromosomes as chroma here to save space. So sex chromosomes are the chromosomes that contain the genes that determine biological sex. And the other type of chromosome that you can have is an autosome. An autosome is just simply a chromosome that is not a sex chromosome. Not a sex chromosome. We have classified these two types of chromosomes, and remember that I said that humans are diploid organisms. And this means that they have two sets of chromosomes. Two sets of chromosomes. Where do these two sets come from? Well, one set comes from dad. I'm going to draw a chromosome here, just schematically, as a elongated rectangle in blue here. And this chromosome is going to be from dad. Let's say it's an autosome. And the other set of chromosomes you get are from mom. This is from mom. So because we get two sets of chromosomes, what this means is that we have two copies of a given gene. So if we map out on this chromosome where a gene might be, and I'm just going to shade it in here. Let's say there's a particular gene and it resides right here on the chromosome that you get from mom. And then the other copy of that gene will reside in a similar location the chromosome that you get from dad. So this is great. We have 2 copies of a particular gene. All right, so that sets the foundation here. And there's one other term that I want to define, and it's what we used to call normal genes. So we call normal genes, generally the ones that aren't mutated. Normal genes are wild type. They're called wild type genes by geneticists. And so we have wild type genes, normal genes. So let's go back and think about HHT. So remember HHT. HHT is an autosomal dominant disorder. This means that the disorder, you only need one bad, say a mutated copy, one bad mutant copy of a gene to have the disorder. Okay, so looking back at those autosomes again. So let's draw that chromosome from dad, blue here. And let's say that dad is the one that gave you your mutated copy, your bad copy of whatever gene it is that causes HHT. And that mom has a normal copy. So let's still go ahead and designate these things. I'm gonna make little h be the wild type gene, and big H I'm going to make the mutated gene. So now we have one bad copy and one normal copy. Whenever we talk about the genetic complement, genetic makeup of an individual, we say that is their genotype. So in this case, the genotype we can simply describe as being big H, little h. That is the genetic complement. That's what copies of this particular gene they have. Now, there's another term that we bring up in genetics, and that's this idea of a phenotype. So what is a phenotype? Well, pheno means to shine or show. So this is how the organism appears. This is what it looks like. And in this case, this would be, say, the symptoms. The symptoms of HHT would describe this person's phenotype, what you can actually see, whereas the genotype is the genetic complement, the genetic makeup of this individual. So along with this idea of autosomal dominant disorder, or how exactly an autosomal dominant disorder leads to a deficit, a dysfunction. And one way is through what's called haploinsufficiency. Haploinsufficiency. Haplo means single. or 1, and insufficiency means not enough. So in this case, this is describing the fact that one normal, and implicit here is the idea that there's a normal gene. So one normal gene is not enough for normal function. You actually need both copies of the gene in order to have normal function. So if you have one that's messed up, the simple fact that it doesn't work means that the other gene can't make up for that loss, and you get some type of defect. The other idea here is the idea of a dominant negative. Dominant negative mutation. And a dominant negative mutation is a mutation where the product of the gene product interferes with the wild type gene. So haploinsufficiency, one normal gene is not enough, and because you're down one good gene, you develop a defect disorder. Or the other idea here is that you can have a dominant negative disorder, and in this case, a dominant negative gene, its product will interfere with the normal wild type gene. And in class, I gave this analogy, and even though you might not be someone who plays video games, it may make a little bit of intuitive sense to you. So I'm going to explain it with that analogy here. So let's say you invited your friend over and you're going to play a game with them on your console. So you're going to need 2 controllers. We're actually going to play in person instead of over the internet. So we need 2 controllers. 2 controllers. And you can think of each controller as a gene in this analogy. So let's say you wanted to play a game. Let's say you play some sort of fighting game. Right? Some sort of fighting game. Those are pretty fun. One of my favorites, Street Fighter, Hadouken, Fireball right there. All right, so you're gonna play this game, and you want to play with your friend. In the case if you had a mutation, let's say, in one of your controllers, and it resulted in haploinsufficiency, you could think of this as this controller here is simply not working. And so since it's not working, you can't actually play the game with the person. So the idea of a dominant negative mutation then is one in that you have both controllers, right? But you have a mutation in one controller, and this mutation happens to cause this controller to send out some weird signal, right? And it sends out a weird signal, and it blocks the ability of the other controller to connect and for you to control your character on the screen. So the sheer fact that you're using this controller inhibits the normal controller, and so it interferes with its function. And that's how you can get a dominant negative effect. So let's go ahead and clear this out. Okay, so to round things out, I want to talk about 2 other terms that are related to the heredity of HHT, and these are the ideas of incomplete penetrance and variable expressivity. So let's use our previous example before. Let's say we had 4 different people, and the genotypes of each of these people is as follows. They each have 1 wild type copy of the gene and 1 mutated copy of the gene that causes hereditary hemorrhagic telangiectasia. All right, so incomplete penetrance means that let's say we had these 4 individuals, and we know their genotype, and when we actually evaluate the individuals, we see that only 3 out of 4 of these people show HHT symptoms. So this 4th one, even though they have the mutation, you can't even tell that they have the disease. This is the idea of incomplete penetrance. How many people that have the mutation actually show the disease? And in this case, 3 out of 4 show the disease. So we say that this hypothetical mutation has a 75% penetrance. Variable expressivity, how's this different? Well, let's look at those 3 people. Let's take those 3 people. We're going to look at them over here. Okay, variable expressivity means that even though these people have the same exact mutation in the gene, they're actually going to show different symptoms from one another. They're not going to present exactly the same way. So let's say this first person here has lots of nosebleeds. Lots of nosebleeds. And that this second person here, well, they have lots of GI bleeding. The first person doesn't really have that much GI bleeding. And then this last person, let's say, this last person has lots of spots on the top of their hands. So the same exact mutation is causing different symptoms in each of the 3 different people, and for this reason, it's often hard to diagnose the disease itself. So with that, we wrap up the first part of the discussion about the genetics of HHT. We talked about some basic principles from chromosomes to genes to diploid organisms. We also talked about the difference between a genotype and a phenotype, how autosomal dominant diseases can manifest as either a haploinsufficiency or a dominant negative effect. And finally, we talked about incomplete penetrance and variable expressivity. So I hope this video helped, and I'll see you in lecture.